Hello, hi everyone. How are you all? I hope you all are doing good, studying for your board exams. They are very much near. I really hope that you're studying and putting a lot of your efforts. Okay, so do remember that this is a time which is never going to come, and try to work hard as much as you can. So today in this video we are going to talk about the TI plasmid, a very small, short and a sweet video, but yes, a very informative video. Okay, so without wasting time, let's get started and talk about TI plasmid. So first of all, what is this TI plasmid? Is it something different from the plasmid which we studied in the previous videos? Yes. Not very different, I would say, but yes, TI plasmid is a plasmid which is a vector for plants. Okay, so this plasmid is isolated from a bacteria called as Agrobacterium tumefaciens. Okay, sorry. So it is isolated from which bacteria? It is isolated from the bacteria that is Agrobacterium tumefaciens or tumefaciens. You can call it as what you want. So where do you find this Agrobacterium tumefaciens? You find it in soil, and this bacteria has an ability to produce ground gall disease in plants. Are you getting it? Are you clear from where you get the TI plasmid? It is found in the bacteria called as Agrobacterium, and this is a bacteria which can cause normally ground gall disorders in plants. Okay, so let us talk more about this TI plasmid. This is the structure of TI plasmid, which you can see it on the screen. So what is the first of all TI? What is the full form is? The full form of TI plasmid is ट्यूमर इंड्यूसिंग प्लाज्मिड. तो उसके नाम में ही क्लियरली पता चल रहा है कि उसका इफेक्ट क्या है, राइट? इट इज अ ट्यूमर इंड्यूसिंग प्लाज्मिड, मींस वो ट्यूमर करेगा. So TI plasmid contains a transposon which is called as tDNA region. See, can you see it is having a tDNA region, right? The jumping gene region. So this tDNA insert copies of itself into the chromosome of infected plant cells. So transposons with the new DNA can still be inserted into the host cell chromosome, and a plant cell containing this DNA can be grown in culture or induced to form a new transgenic plant. Now, what is it exactly? Let us try to understand that. See, this bacteria is usually a bacteria which causes ground gall disorder. So how will you prevent that? That I'm going to show you with the schematic, and then you will understand what was written on the screen in the last slides. See, Agrobacterium tumefaciens is a bacteria which is found in soil. It's a tumor-inducing bacteria and causes gall tumors in dicotyledonous plants. So it looks something like this. Okay, the gall looks something like this. Okay. So now understand this concept. This is a rhizobium bacteria. This is an agrobacterium bacteria. So what you have to do, you have to first isolate the plasmid. Okay, दोनों का भी plasmid is being isolated. Agrobacterium के plasmid का नाम भी know, that is TI plasmid, right? So now we have to cut both of these plasmid with the restriction endonuclease enzyme. Okay, here you are going to take the tDNA cut region, and here you are going to take the NIF. And this have to be ligated by using a DNA ligase enzyme, and you get a new recombinant plasmid which can be inserted into the again agrobacterium cell. Okay, and the cell which is going to accept this new recombinant DNA is called as transformed cell. So now what we have to do, we are done with the recombinant technique. So now we want the multiplication of these kind of the cells. So this recombinant cell is being multiplied and the multiple copies are being obtained. Can you see? A lot of multiple copies are being obtained, but this is carrying the rDNA. That is the recombinant DNA. It is carrying which DNA? It is carrying the recombinant DNA. So now what we have to do? We have to insert this recombinant DNA, the recombinant DNA plasmid which has in the cell, that is the agrobacterium cell. And now what it will do, it will silence this region and it will prevent the gall disease. Okay, it will prevent the gall disease. Okay, so I hope you have understood the entire mechanism. So normally this Agrobacterium tumefaciens is a bacteria which has an ability to cause this disease. Okay, so what are we doing is we are going to see that we form a new bacteria, that is we form a new recombinant uh, plasmid, which will not cause disease in the plants. Okay, so this is the technique how you can form a recombinant DNA, and then you can introduce into the plant so that the plants are prevented from these diseases. So this bacteria can be used for BT crops, transgenic plants, and genetically modified plants. We will be talking in detail about BT. What are BT? BT, the full form of BT is Bacillus thuringiensis, which is also one of the very important mechanism for your exams, which we are going to talk in detail in one of the videos. Okay, so that's all about the TI plasmid. Very simple. From which bacteria it is obtained, you should know. That's it. Nothing very difficult. So I'll meet you soon in the next video. Till then, bye. Take care. Work hard for your dreams.